Hey guys, I'm here. I know there's usually a delay, so I'm not sure when you're going to be able to start hearing me, but I'm here. Um, so what I need you to do um, is just write in your question where you can probably find the chat window um, um, where there's a menu, there's a drop down, there's a chat window. So you can chat in your questions, and then as soon as I see some questions, then I will answer them. In the meantime, I will wait. So if you can hear me, if you could just confirm that. It looks like my mic is picking up my voice, but if you could confirm that you hear me, that way I don't look like an idiot and you can just see my lips moving and nothing's coming out. I know there's a lot of sarcastic comments that could come from that, but we're not going to go there. So, like I said, I don't know what kind of a delay it's on. Hopefully you all can hear me. But as soon as you can hear me, if you could somebody just chat in to say that you can hear me or see me or something. All right. Somebody says it's supposed to be starting at 6. I'm here. I hope you can see me and hear me. So somebody, please, I hear you and see you. Okay, good, good. Armando, I wonder, is that you, Armando? AXR. I'm trying to think of who else it would be. Okay, okay, good. Okay, so we went over most of the review today in class. So what we didn't review was weathering, erosion, and deposition. I would rather go over the questions that you have on the stuff we already went over, because it kind of is silly for me to go over weathering, erosion, and deposition if I'm going to do that tomorrow. So do you have any questions about what we talked about in class today? Let's see, I was just finishing the test. Hang on. Here it is. I'm going to have to make a lot of changes from the past years because we didn't get as far. Because we did Texas ecoregions. So I'm going to have to make some changes to it. Hang on. I have to go back to the screen. There it is. All right, so like I said, I don't see questions. So make sure that you chat in questions so that I can answer them. But going over the review, basically the kinds of questions that you're going to get, like when it comes to equipment, it would be something like, Susie wants to get the mass of a penny. What tool would she use? Ah, sorry, Milo, that's my dog. What tool would she use? And the choices would be, graduated cylinder, beaker, triple beam balance, bathroom scale. And then you would need to, hang on, get off my bag. And then you would need to tell me triple beam balance, because that's what you would use to get the mass. So that's how we would get the equipment questions. And then somebody said that fermentation, oh, Grace, I think, fermentation wasn't on the review. I know. It should have been, so, but since it wasn't, it's not going to be on the midterm. So I can't put it on the midterm if it wasn't on the review, because I told you I wouldn't do that. So the test is multiple choice, all multiple choice questions, 50 multiple choice questions. So you have to be prepared to come in and get to work right away, because you will only have the class period to answer the questions. You won't have extra time. What's the product and the reaction? Those just fancy words for the product is what the process makes. So the products of photosynthesis are glucose and oxygen, and the products of cellular respiration are ATP. Hey, easy. Carbon dioxide and water. So product is just what the process makes. Remember, in a multiplication problem, hang on, honey. In a multiplication problem, the product is the answer. So you can help you remember that way. And then reactants is what it starts with. So that's reactant and product. Kai, I don't understand fermentation. You can come in and we can talk about it, but it's not on the midterm because I didn't put it on the review. So I'm not going to take the time to go over it right now. I wished I had put it on the review because we did spend quite a bit of time on it. So it's not on the test. I don't get liquid and solid volume. All right, well, when liquid volume is when you're getting the volume of a liquid like water or oil or I don't know, something that's liquid. You get the volume in a certain way. And then if you solid volume is getting the volume of something that isn't a liquid, like a cube or a pebble or a marble. So liquid and solid volume is just the volume of a liquid or the volume of a solid. And then how you get that is a liquid, obviously you pour it into a graduated cylinder of some sort. And then a solid, if it's a regular solid, you'll do length times width times height, which means the unit of measure will be centimeters cubed. But if it's an irregular solid, you'll do water displacement. And that's when you drop the item into a graduated cylinder that's at a certain level. So say you have a graduated cylinder that you add 20 milliliters of water to, and then you drop a pebble in and it goes up to 24. You subtract 20 from 24 and you get 4. So the volume of that irregular solid is 4 milliliters. You could also say it's 4 centimeters cubed because 1 milliliter equals 1 centimeter cubed. So liquid and solid volume is just the volume of a liquid or a solid. There's a question that if you don't finish, you have to finish. You can't come back. You have to finish. So that's why you've got to get in there and get started. There is no extra time. You can't come after school. You've already seen the exam, so you can't come back and finish it. Could you go over osmosis and diffusion? Okay. So osmosis, well, let's diffusion first. Diffusion is the spreading out of molecules going from an area of high concentration to an area of low concentration. So my aroma diffuser in my room, the thing that sprays out the smelly stuff, if you stand when I first turn it on and you stand really close to it, you can smell it really strongly, but from far away you can't smell it. That's because those excuse me, those particles are really close together, but they're eventually going to spread out. So the scent is really strong up close, but then they spread out. So they're going from an area of high concentration, which is where there's a lot of them, to a low concentration where there aren't that many. The goal is for the molecules to reach equilibrium or homeostasis, where there's an equal number of molecules throughout the entire room. Another example of diffusion is if you put food coloring in water. So you drop it in the water and it's really concentrated in the center. You can almost see like a line of food coloring. And then if you watch it, even if you don't shake it, you watch it, all those molecules spread out. And that's diffusion. That's that water, that the food coloring molecules moving from an area of high concentration where they're tight, close together to low, to spread out until they're equal. And then osmosis is the diffusion of water across a semi-permeable membrane. So the diffusion of water, so water going from an area of high concentration to an area of low concentration. Stop. Hey, quit it. Sorry. Water osmosis, diffusion of water across a semi-permeable membrane. So the difference between osmosis and diffusion is osmosis has to cross that membrane. Diffusion doesn't, but osmosis does, and it's water. So the diffusion of water across a semi-permeable membrane. Do we need to know Texas ecoregions? No, you do not. I do not understand water displacement. Okay, so I think I just explained it, but just in case, it's how you get the volume of an irregular solid. So if I wanted the volume of my hand sanitizer, oh, geez, sorry, my dog's a little relentless with the petting. Stop. So hand sanitizer. Well, Luna, stop. Sorry. Hand sanitizer. So I can't get length times width times height of this. Wait, there we go. Because it's irregular. It's not a regular shape. I could do this pretty much because it's a regular shape, right? But I can't do that. So I would find a beaker big enough that I could drop it in, and I would put the beaker at like 20 milliliters. And then I would drop this in and see how much the water went up. The water might go up to say 31. So it went from 20 to 31. I would subtract and I'd get 11. 31 minus 20 is 11. So the volume of my hand sanitizer would be 11 milliliters. Now remember, volume is, it's interesting. It says on here 29 milliliters. Okay, but that's how much this container can hold. But that is not like, that doesn't include like that. Volume is how much space something takes up. So the milliliters only counts for the stuff inside, not the container. So just in case you're wondering. All right. Okay. Let's see. Can I explain excretion? Well, excretion is getting rid of waste. So there are different organs in your body that do that. The first ones are obvious. It would be your large intestine or your rectum or whatever you want to call it. And your bladder, that would get rid of liquid and solid waste. But then our skin also gets rid of waste, sweat and things like that. Our lungs get rid of waste, which is carbon dioxide. So excretion is getting rid of waste. Okay, so that's one of the life processes. Do we need to memorize the 7 life processes? Yes, you do, and it's Mrs. Gren, MRS, and then GREN. That's the foldable in your interactive notebook. So just remember, it's a mnemonic, so that means each letter stands for one of the 7 life processes. So M is movement, R, respiration or reproduction. And also respiration doesn't necessarily mean breathing. So when you think of a plant, it's not breathing the way you would think of breathing, breathing air in and out. Respiration means the exchange of gases. So respiration S would be stimulus or sensitivity. G is growth. The other R is respiration or reproduction. E is excretion and N is nutrients, needs some sort of nutrients. Yes, you need to memorize those because it might say which one of these is not is necessary for life or is not one of the life processes. That's how that question might be worded. What is energy from photosynthesis stored in a plant as? Glucose. Sorry, plant energy is glucose. It's very important. Well, let's see. How will questions on converting measurements be formatted? It'll be multiple choice. Like the questions on the review, it might say something like convert 58 grams to how many milligrams, and then you'll have 4 choices, and that's how you'll have to figure out. Okay, 7 life processes. I just went over that. I don't understand lactic acid fermentation and alcohol fermentation. It is not on the quiz. I mean, excuse me, the midterm, so I'm not going to go over it right now. So fermentation was not on the review, so it's not going to be on the test. Okay. Will you explain passive and active transport? Okay, so sorry, my throat's really itchy. Passive and active transport. So we're talking specifically about transport across a semi-permeable membrane. So passive transport means that movement across a semi-permeable membrane does not require energy. And an example of that is osmosis, which is when the particles cross the membrane automatically because they want to be equal. So little water molecule comes up, gets to a membrane, it's like, hey, there's more of us out here than there are inside. Let me in. And the cell membrane goes, okay, and it goes in because it's not equal. That's passive transport. Active transport is when energy is required for some reason. And there's 2 types of active transport. There's engulfing, which is where amoebas absorb. They just kind of go and engulf their food because that's the only way amoebas have to eat. And then there's transport proteins, and they're like little pathways that these transport proteins create in the cell membrane so that molecules can pass. And there's a couple of reasons why the molecules would need to pass or need help passing. One is the molecule is simply too big. So if you are at the playground with your little brother or sister and you want to, sometimes when you get older you can't fit into the little holes anymore like they used to climb through, and hopefully you won't get stuck. Thank good God that's never happened to me, but you're too big to fit through the hole. So the only way to fit through is for somebody to shove you through or for them to make a bigger hole. So it's the same thing with big molecules. Glucose molecules are huge, so they can't pass through a membrane without help. So that help is a transport protein. Also, if there are plant cells like cactus that have to store water because they anticipate having a drought or whatever, then sometimes there has to be more water inside the plant cell than outside the plant cell. And that's when they might need help crossing the membrane because it won't happen automatically because it's not equal. It only happens without energy when it's trying to be equal. All right. Will you explain how to measure a liquid? To measure a liquid, you have to use a graduated cylinder. So if I have my, oh, it's even better, I have my Diet Coke, and I wanted to know how much the volume was, I'd have to, well, I could cheat and just look at the outside of the bottle, 355 milliliters, or the can, but then you have to pour it into a graduated cylinder, something that has measurements on it. So a question might be, Susie wants to get the volume of her Diet Coke. What tool would she use? And you might get triple beam balance, a beaker, graduated cylinder, centimeter ruler, and you would say triple beam balance, graduated cylinder. You always measure liquid volume with a graduated cylinder. Now, measuring the mass of a liquid is different because you may remember us doing that lab where we talked about the density of water. Technically, the volume, like 1 milliliter equals 1 centimeter cubed equals 1 gram. That is technically true. But if you don't have access to a graduated cylinder and you want to get the mass, you would have to use a triple beam balance. But don't forget that you have to measure the container that it's in first and then add the liquid and then subtract the mass of the container, because then you would be getting the mass of both. So you might be asked a question like, what tool would you use? But beyond that, because there aren't any short answer questions, I wouldn't worry about the rest of it. I don't understand lactic acid fermentation. We're not doing that, so you don't have to worry about it. On the test for water displacement, how will the questions be formatted? So you might have a question like, Susie, I always say that name, I have no idea why, was trying to find the volume of Diet Coke, and no, that wouldn't work. Water displacement. See, I need this Diet Coke really bad. That's why I can't talk about anything else. All right. Susie wanted to find the volume of her hand sanitizer container. So she placed it in a graduated cylinder that was originally at 20 milliliters. The water rose to 50 milliliters. What is the volume of the hand sanitizer? And then you'd have 4 questions. Will you explain how to convert measurements? That's that whole stair-step thing. And I don't have that's one of the downfalls of this, the Ustream, is I don't have like a screen thing, or at least I don't know how to do it. Hang on. All right, so King Henry died by drinking chocolate milk. Nah. Okay, so King Henry died by drinking chocolate milk. So I did my stair step and I used that mnemonic. Oh, other side. King Henry died by drinking chocolate milk. And this is kilo, hecto, deca, base, deci, centi, milli. And that's the same regardless of the base unit. Okay? Kilo, hecto, deca, base, deci, centi, milli. So, and base is meter, liter, gram. All right? So if you get a problem like convert 55 grams to how many kilograms? So I'd have to, where am I starting? I'm starting at, sorry, this is backwards. I'm starting at grams and I want to go to kilograms. So I start at base and I have to go to kilo, because that's the prefix. So you go 1, 2, 3. Remember your decimal point is right there. Okay, so it would be 55 and I move it up 3, which means I move it to the left 3. So I go 1, 2, 3. Okay, so you just get that stair step. I've always found that the easiest way. And then if you move up the stairs, you go to the left. If you move down the stairs, you go to the right. Describe diffusion. Okay, I already have. but I will do it again. Diffusion is the movement of molecules from an area of high concentration to low concentration. So I have my little aroma diffuser, and then it spreads out. Okay, whoa. Okay, so see it's really concentrated right here, and then it spreads out because it's going from where it's really strong, highly concentrated, to where it's not so concentrated, low concentration. So you can see all these molecules are close together. That's low concentration. No, sorry, that's high concentration. Wow. I'm more tired than I thought. Concentration. And then this is low concentration. And you can see why, because, oh my goodness, the little dots are really close together here. Right? But they're not, they're almost non-existent out here. So that's low concentration. Remember, concentration is like when you have lemonade concentrate and it's frozen and it's really strong. And if you just put that in a cup and drank it, it would be, I don't know. Sometimes I like to add a teaspoon of it to like some carbonated water or something, but it's really strong because it's concentrated. So then you add water and the molecules spread out, just like Gatorade concentrate. It's powder that is concentrated, it's really strong, and then when you add water, the molecules spread out and it becomes equally distributed and it tastes better. All right, is it all multiple choice? Yes, the whole thing is multiple choice. Could you explain cellular respiration? All right, cellular respiration is when energy is necessary, but there isn't enough oxygen. No, that's fermentation. I was reading another question. Cellular respiration is how plant and animal cells get their energy. Wow, I'm just tired. Cellular respiration is how animal cells get their energy. So they start with glucose and oxygen because that's what plants give off. So we start with glucose and oxygen because we breathe in oxygen, and glucose is what our food breaks down to into glucose molecules, and then that makes carbon dioxide, which is a waste product that we breathe out, water, which we need in our bodies. Our blood has water, our muscles, our skin, we have a lot of water in our bodies, and ATP. And ATP is animal cell energy, okay? So cellular respiration is how animal cells make their energy. Osmosis. Again, osmosis is the diffusion of water across a semi-permeable membrane. So you need to know the difference between osmosis and diffusion. And osmosis is the diffusion of water. But you have to say also across a semi-permeable membrane, because if you don't say across a semi-permeable membrane, it's not going to be correct. Okay, what does a vacuole do? A vacuole stores water, nutrients, and some waste, mostly water, especially in a plant cell. So when you water your plants, you're actually filling up their vacuoles. So when the vacuole runs out of water, the plant wilts. And when you water the plant, it perks back up again. Vacuoles actually give, in addition to the cell wall, they actually give plant cells a lot of their shape because they are so big. So if a vacuole shrivels up, the whole plant cell is going to kind of, but when the vacuole is full, it's perky and your plant looks better. I don't understand isotonic. We're not doing isotonic, hyper, and hypotonic on the midterm, so don't worry about it. Can you go over independent and dependent variables? Okay. Independent variable is what a scientist changes in an experiment. So Let's say I want to see if, I'm trying to think of something other than my plant one because I use it all the time. Let's see, how about, okay, I just made cornbread. My kitchen smells really good. So I wanted to test if one sort of mix, no, how about the spray, the pan, like you have to put oil in the pan? How about if I just test whether the spray pan is better or putting like shortening inside the pan is better? So that would be my test. I'm going to treat the pan differently. I'm going to prepare it differently. So my independent variable is what I changed in that experiment, okay? And in this case, it would be the treatment of the pan before I cook it. So whatever sort of oil or whatever I put in the pan first. So that's an independent variable. So then the dependent variable is how much the cake or the cornbread sticks to the pan, how easy it is to get it out of the pan. So then I have to come up with a hypothesis. So I would say if a pan is sprayed with PAM, then it will come out of the pan easier than if shortening was used. And then I have to test it. So the independent variable is the type of treatment of the pan, and the dependent variable is how easy it comes out of the pan. I don't know how much baking you guys have done, but I can tell you it's a really big drag when you're making something and it sticks to the inside of the pan. So that would be my dependent variable, would be how easy it comes out of the pan. So then the control group would have to be what I usually use. So I usually use PAM. So that would be my control group because I was comparing it to the shortening, or vice versa. You could go either way with the control group. You have to decide. All right. So in this case, control group that gets no special treatment. Let's see. So in this case, the special treatment, let's say, would be the PAM. So that would be the control group. And then the constants would have to be the cornbread mix, or the ingredients for the cornbread, the type of pan, how long it was cooked for, blah, blah, blah. So independent variable is what we as scientists change, and the dependent variable is what changes as a result of that independent variable. All right. What takes place during photosynthesis? Really? What takes place during photosynthesis? Plants make energy. Plants make their cell energy. That's what they do. They make glucose. Is it called osmosis when oxygen crosses a semi-permeable membrane? No, it is when water crosses a semi-permeable membrane, not oxygen. Semi-permeable is a free membrane, right? Selectively permeable membrane is only certain things go through. No, selectively permeable and semi-permeable are the same thing. To be semi means partially, and selectively also means partially. So those words are used simultaneously. Semi-permeable and selectively permeable mean the same thing. Does the cell wall give the shape for plants? Absolutely. That is more so than the vacuole. I was just saying that vacuoles also tend to give plant cells their shape, but that's the cell wall. The most important things that it does is give the plant cells its shape and protection. I need to go. Can I watch it later? Yes, it'll be posted on Blackboard about 15 minutes after it's over, which will be about 7:15. Okay. What is the difference between mitochondria and chloroplasts? Mitochondria is where cellular respiration takes place in an animal cell, and the chloroplast is where photosynthesis takes place in a plant cell. Animal cells and plant cells both have mitochondria. Mitochondria are a lot smaller in a plant cell, but plant cells and animal cells don't have chloroplasts, only plants do. And chloroplasts, again, chloroplast is where photosynthesis takes place, and mitochondria is where cellular respiration takes place. Okay, will you help me remember what the cell organelles in the cell? I'm not sure your question, so if you could rephrase that, will you help me remember what the cell organelles are in the cell? I don't know how to help you memorize that. We've done a lot of activities that, you know, have you compare cell organelles to the school or things like that. Because it's multiple choice, you're not going to be asked to list the cell organelles, but you are going to have to know the functions, and that's why we've been doing so many different activities with that. So I don't know how to help you memorize it, because we just went over it in so many different ways. There's no mnemonic device. You've just got to do it. There will be a diagram on the test where an organelle will be pointed to, and it will ask what's its structure and function. Are the 4 causes of erosion wind, water, ice, and gravity? Yes, but we're not going to talk about those tonight because I'm going to go over them tomorrow. But yes, it's wind, water, ice, and gravity. Also, boys and girls, remember that Blackboard, there is a folder for everything that we've learned, and there's a lot of information in each folder, including the PowerPoints that we saw, the notes. So make sure that you use those resources, because it's silly to try to guess at this information. It's a lot of information. Okay, the difference between a control and a constant. That's a good question. That's one that comes up a lot. Control group is the thing that gets no special treatment. Okay? A constant is the variables that say constant or the same for all groups. So how I remember it is the constant, another word for constant is same. So what things have to stay the same. So control is a control group, the group that gets no special treatment, and a constant is what variables are the same for all groups. I keep forgetting the lysosome. Is there a memory trick? Well, how I always remembered it is lysosome reminds me of Lysol. And that you might not be able to remember that. I don't know how much cleaning you do with Lysol. It actually smells pretty gross. But it's a disinfectant, and the lysosome is in charge of cleaning up the cell. So that's how I remember it, as a lysosome. I think of Lysol. Is the information from the web searches in WAD unit going to be asked on the test? The questions that are going to be asked on the test are: what are the causes of erosion, weathering, erosion, and deposition? You know, if sediment is put down in a new place, what is that an example of? That kind of thing. And yes, the whole thing is multiple choice. You don't have time for short answer. So yeah, the whole thing's multiple choice. You guys have all got to be better about keeping track of your work and putting everything in your interactive notebook. I go through some of those notebooks and I see a lot of students have been very conscientious about it. But so that what happens is when they go into, oh, my dogs are wrestling. When they go in to find their work, it's all there. So like, where are my notes for this, or what's the answer to such and such? You can just open your interactive notebook, hey, and it's there. But, ow, stop, no, Luna, stop. But if you have all your stuff spread out all over the place, it's going to be very hard to keep track of everything. What is the lysosome? Oh, Evan. Okay, maybe you had an ad while I was explaining it just now. But a lysosome is responsible for cleaning up the cell. So it packages up all the waste and moves it to the outside of the cell. If the waste gets to be too much and the lysosome explodes, then the cell dies. Remember that cells are dividing and dying at an incredible rate in your body all the time. So don't panic that when I say your cell will die, because it's sorry, it happens all the time. Is there an answer sheet to write on, or circle the answer on the test? It's an answer sheet. You cannot write on the test. Are we going to be asked differences between prokaryote and eukaryote and animal and plant cell? Animal and plant cell, yes. Prokaryote and eukaryote, no. Is cell theory the guy who first figured out what the cell was? No. Cell theory is three things. The first is all cells come from pre-existing cells. Cells are the smallest units of life, and everything is made of cells. Those are the three parts to cell theory. Those are also in your notes. And let me check. I'm pretty sure, I can't see you right now. I'm pretty sure they're on Blackboard. I organized everything the other day. It was just kind of organized by whenever I loaded it, which was not effective at all. Hang on. Okay, pre-AP. What was I talking? Oh, cell theory. All right. Cell structure and function. What? Wow. The filled in notes for cell structure and function are on Blackboard. All living things are made up of cells. Cells are the smallest working units in all living things. All cells come from pre-existing cells through cell division. It's all on there. But no, we will not talk about eukaryote and prokaryotes. So use your resources, people. Do we need to review things from Texas ecoregions? No. Please remember, if it's not on the review, it's not on the test. I promise I am not trying to trick you or anything, really. There's nothing on the test that is not on the review. I wanted fermentation on the test, but it was left off of the review, so I took it off the test. So I am not, myself, Miss Stokes, Coach Bauer, we are not out to get you. We are just testing your mastery of the information. And that's what we have to do. So, you know, when you get to college, you might end up with a class that only has 2 assignments all semester, a midterm and a final. And your ability to take a test and study and process all this information, you might just have 2 grades in the grade book, and that's tough, because if you tank one test, you're kind of in trouble. So fortunately, we don't do that in middle school. Let's see. So let's see. There's a lot of other things on Blackboard. Let's see. Science process. Oh, there's practice on there. The cards are on there that we have done in the past. Those yellow pieces of paper that are in the room that we haven't had a lot of free time, but that's something you're supposed to be doing with free time. Remember, with the folders. So science process skills, I'm looking at them right now. Qualitative versus quantitative. Let's see, observations versus inferences. Variables, that's all on Blackboard under science process. Will there be any, okay. Do we have to know the numbers in the formula for photosynthesis and cellular respiration? If you are in pre-AP, which is 2nd and 7th, you will have to know the words and the numbers and letters. If you are on regular, you just have to know the words. And when I say the words, I mean this. So carbon dioxide, my pen is squeaky, yields, bloop, 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 sun, glucose, and oxygen. Those are words. Okay? That is what regular or on level needs to know. Carbon dioxide plus water yields, with the sun, glucose plus oxygen. Pre-AP needs to know this. Carbon dioxide, it's 6 CO2 plus 6 H2O yields, with the sun, C6H12O6 plus 6O2. So, pre-AP, you have to know both. Regular, you have to know the top. Now, do not, well, you're not going to be writing it, but this is not the words. I had somebody do this a couple people do this on the quiz. 6 carbon dioxide. That's not what I'm talking about when I talk about the words. I'm just talking about that. All right. How important is the grade? What happens if you don't do well? Yes, it'll affect you. It is, somebody said 1/7 of your grade for the year. It's actually 1/8, because you have the 3 6-weeks grades and then your midterm grade. So it's like having 4 6-weeks grades. And then you have the next semester, which is 3 6-weeks, plus the final, which is a 4th 6 weeks. So it's like 4 plus 4, it's 8 grades. So the midterm is 1/8 of your whole year grade, and the final is 1/8. And the final is not cumulative from August, it's cumulative from the day after the test. So Thursday is when we start new stuff. Do we need to prepare for any questions that will have us break down an already performed experiment? Well, you will definitely be given scenarios. So it might be so-and-so completed an experiment where, and then they'll give you a scenario, and then what is the independent variable in this experiment. So yeah, I mean, that's the only way to do it, because it's multiple choice. So There's no way for us to ask you to write an experiment or write a good hypothesis or graph something. So absolutely, you will have to break down an experiment and give, like I said, what is the control, what is the constant, all that stuff. Hi, honey. Okay, it says, do you give the exact same questions from the review? The review isn't it technically questions? I mean, it is. But when it says explain photosynthesis, that's not a question. So no, it's not the same. The review goes over concepts. It does not go over it question by question. So no, it is not going to look like that at all. But you do, okay, but you will have to know the concepts. So you have to know cellular respiration, photosynthesis, cell organelles, all that stuff. You're gonna have to know it. You don't do the final at the end of the year if you get an average of 85 or above. No. I don't know where you got that, Sandra, but you don't do the final at the end of the year if you get an average of 85 or above in the 4th and 5th weeks. That is not true. That may be true in high school. It is not true in middle school. So you take the final regardless of your grade. How many questions are there? There are 50 questions. 50. I've said that a lot of times. There are 50 questions. I really don't want to answer questions about the exam anymore. It's important. You have to study for it. If you don't do well, but you've done really well over the other 6 weeks, it won't kill your grade. If you are on the verge of not passing and you don't do well on the exam, then you're going to be in trouble. But I'm not going to answer any more questions about the exam specifically. Yes, it says, will there be questions where we have to identify a cell organelle? Yes. There will be questions where there is a diagram of an animal cell or a plant cell with an arrow pointing to it. It'll ask, what is this organelle? And then the next question might be, what is its function? So yes, there will definitely be questions like that. Let's see. So as soon as we get off here, I am going to post the answers to the review onto Blackboard. It'll be in that folder under midterm or exam resources or something like that. In addition, I will finish this broadcast and also post that into Blackboard. Also, I found the last video that I did, which apparently I had laryngitis, but the last video I did on Ustream, one of the videos that's still on Ustream is on cell functions, structure and functions, and actually on cellular respiration and photosynthesis. So if you want to listen to it, you can. It was weird for me to watch myself talk, so, but I could tell that that's what it was on. If you have any other questions, please make sure you mark up your review, and then you ask them tomorrow in class, and we're going to go over weathering, erosion, and deposition. So if that's it for the questions, I'll wait another 30 seconds or so, but then I'm going to stop the broadcast and load all that other stuff. Will there be questions where we have to convert measurements? Yes, there will. So I'm going to end the tutoring session now. Again, if you have any other questions, make sure you bring them to class tomorrow. I'll have tutoring after school, before and after school tomorrow. Take advantage of the resources that are there for you. I know you have other exams, but there are some that you can just watch some videos or whatever. So if you have any questions, just make sure that you ask them tomorrow. And I'll see you guys all tomorrow. Bye.